Our last video just ended with glycolysis. Let's write that pathway out for you here, and we'll see how well all of this new technology stuff goes. Okay, so let's start, and I'll draw both longhand version and essentially the shorthand version of carbohydrate catabolism as we go through this. Let me expand this a bit. Okay, so for glycolysis, and we're writing out the pathway of aerobic respiration. Using carbohydrates, carbohydrate. Catabolism. Closer, so I'm close to the screen. So starting with glucose as our initial compound. The glucose is a 6-carbon, 12 hydrogen, 6 oxygen compound. Now when this is transported into the cell, right, glucose is outside, and it's transported into the cell through a glucose permease molecule, and it's modified as it goes through. This is going to be our shorthand version. So we'll just say this first process that we just saw is called glycolysis. The end products of glycolysis after partial oxidation of glucose are 2 pyruvates or pyruvic acids. These are 3-carbon compounds. Now in this process we have ATP, right? So a net total of 2 ATP generated via substrate level phosphorylation. Right, there are 4 that are used, there are 2 that are used, there are 4 that are produced, and therefore a net total of 2 ATP through substrate level phosphorylation. There is also reduced electron carriers generated here. There are 2 reduced NAD molecules to generate 2 NADH plus H plus. And this is our net yield here for glycolysis. Now we're going to write over here our longhand version of the same process. See if I can move my toolbar. Okay, the longhand version of it, I'm going to write over here, the exact same process. So here we have glucose as our starting compound. Again, it's brought into the cell. And through a series of reactions, we end up with a 6-carbon diphosphate. And I'm going to indicate some of the different things that are happening. Oh, find my colors down here. Oh, not pink. 6-carbon diphosphate. And whoops, wish there was a way that I can undo that. Oh, undo, haha. Okay, and these phosphates come from 2 phosphate transfers from ATP molecules, 2 of them. They donate their phosphates to make 2 lower energy ADP compounds, right? So this is that input of 2 ATPs to phosphorylate this molecule. And there is a diverging event that occurs here. Where did I go? Here, where this molecule splits in half. Move my little face. There. Where am I? Okay. So this molecule diverges here to generate two 3-carbon compounds that are still phosphorylated. There's a 3-carbon compound here, a 3-carbon compound here. Both of these have a phosphate on them. And then from here we have our 2 linear pathways. To generate our 2 3-carbon pyruvates. Three carbon, one of them here and another one here. Now there are some phosphorylation events that are occurring. So there's an independent phosphorylation of this 3-carbon compound, and therefore there are 2 phosphates on each of those guys that are able to phosphorylate ADP. 2 of them here through substrate level phosphorylation and make 2 high energy ATP. Right? And same over here, 2 ADP are phosphorylated on this linear pathway to generate 2 energy-rich ATP compounds. There's also a reduction event that is occurring. There is a reduction event occurring on each of these guys, on each of these linear compound or linear pathways. So here's one to generate 1 reduced electron carrier. And here's another. All right, and that's really all that you need to know for glycolysis. Now let's step back over to the slides. Hopefully, I think this whiteboard will still stay here. Hello. All right. So this is the yield for glycolysis. We have 2 ATP gained in this process through the process of substrate level phosphorylation. Of course, 4 are produced, 2 are consumed to make 2 total, or 2 net total. There's 2 NADH plus H plus generated in glycolytic pathway, and then there are precursor metabolites that are generated at each step along the way. All of these can be pulled out at any time and used for other processes in the cell, specifically biosynthesis processes, but we are going to continue working through what happens with pyruvic acid at the end of this pathway and watch the complete oxidation of this compound. Now, note though, that pyruvic acid has many different venues that it can travel. So pyruvic acid can enter into the Krebs cycle, which we will follow through here in just a minute, to complete the process of respiration. In other words, the complete oxidation of glucose. But alternately, this molecule can be used to essentially just re-shuttle those NAD electron carriers in the fermentation pathway through either alcohol or acid-based fermentation. And this would just be a very rapid shuttling of those electron carriers to help produce ATP through substrate level phosphorylation of glycolysis for those organisms needing to do a very, very rapid turnover of glucose. And then alternately altogether, this molecule can be used for anabolic processes. So this can be a key ingredient in amino acids. This is actually the basic center compound for amino acids, for different carbohydrates, and also for fatty acids. So this is a really important metabolic intermediate. Here's a picture showing all of those different pathways. We just looked at glycolysis and how glycolysis ends with this pyruvic acid 3-carbon compound. Note that there are still 3 oxygens on here, but instead of our 6 hydrogens, now there are only 4 because 2 of them were lost in those reduction oxidation events that we saw in glycolysis. Here are those biosynthesis pathways I just mentioned. So amino acids, sugars, or fat metabolites can be generated here through biosynthesis. There's many, many pathways involved in that. This molecule can alternately be immediately converted into lactic acid by becoming oxidized. So those hydrogens can be donated back onto this molecule to generate lactic acid, or to generate, so there's alterations to this to generate acetaldehyde, which then can receive those 2 electrons and 2 protons to become isopropanol, and then alternate alcohols as well. So these are our fermentation pathways. But we're going to focus here where pyruvic acid is shuttled through or is transitioned into an acetyl group picked up by a coenzyme A, and then this is donated to the starting compound oxaloacetate in the Krebs cycle to turn that Krebs cycle one time and complete the oxidation of this compound for aerobic respiration. So in order to continue this process, there is first an oxidation event that occurs, oxidation and reduction event, where that 3-carbon pyruvic acid is converted into a 2-carbon acetyl group that is picked up by a coenzyme A. So this is that transition step. Let's look, I think I can just go directly there. Let's look at that whiteboard, and we'll draw this out on both sides. So we'll change, whoops. Here we go. Over here on our shorthand version, this is our transition step. Where 2 carbons of that 3-carbon compound are picked up to form an acetyl group. And we're making 2 of these. That acetyl group is picked up by a coenzyme A. Now remember, we're doing this shorthand. So coenzyme A, that should be lowercase. Let me fix that real quick. Coenzyme, I'll just write that out. Coenzyme A. These guys swoop in and pick up that 2-carbon acetyl group. So now we have acetyl coenzyme A. This is the apo enzyme, and that's the holoenzyme, if you remember those terms. Now we also have a reduction event occurring and an oxidation happening. So carbon dioxide is lost. So that 3-carbon, there's only a 2-carbon molecule picked up here. So one of them is released as carbon dioxide. Now I'm showing you what's happening for every input of glucose. So there are 2 pyruvates generated, therefore this is occurring 2 times. So I'll indicate that by saying 2 CO2s are lost here. And we also, of course, with a reduction, with an oxidation event, this is coupled with a reduction event. So here's our reduction event where NAD is reduced, and for every 1 pyruvic acid, there's 1 NAD reduced. So we'll indicate that there are 2 events occurring here because of both of those pyruvic acids. So there's our 2 reduced NADH plus H+. Let's draw that over here for our longhand version. And this becomes important when we are trying to do, oops. This is gonna work. All right, this becomes important to do this longhand version when we're trying to understand what's happening when we are using a non-carbohydrate starting compound. So again, this is our transition step where we have an acetyl group, a 2-carbon acetyl group that's generated through a reduction oxidation event of that pyruvic acid. So it's oxidation of the pyruvic acid. And that oxidation event is coupled with the donation of that coenzyme A. I'll write it over here. So for each pyruvic acid, there is 1 coenzyme A that comes and picks up that acetyl group to form acetyl coenzyme A. That's happening here, and it's happening here to form an acetyl coenzyme A. And again, this is an oxidation of pyruvic acid, an oxidation where we have a loss of a CO2 compound. Here, a loss of a CO2 compound and a reduction of an NAD. And same over here. Okay. So here we are, we've just transitioned, and again, we just saw all of this stuff. So let's watch the destination of that acetyl coenzyme A. This compound will enter into the Krebs cycle. The Krebs cycle is a cyclical metabolic pathway. So those hydrogens are eventually downstream transferred to oxygen. So those hydrogens need to go from that acetyl group generated from that pyruvic acid to be donated to electron carriers so that ultimately they can be converted into energy. This is all occurring in the cytoplasm of bacteria in the mitochondrial matrix of eukaryotic cells. And there is a significant generation of reduced electron carriers here, both in the form of NAD and FADH2. There's also a small amount of ATP generated for each turn of the Krebs cycle. Overall, it looks like this. I'm going to have you pause here and pause my video, go into our lecture videos and watch this, how the Krebs cycle works from your lecture videos. I don't need to waste our time here on it, so you have access to that one on your own. But let's walk through this process on our whiteboard. Okay, so let's do our longhand version first, because I think it's nice to watch what's happening. So this 2-carbon acetyl group will be donated to the first molecule in this Krebs cycle. I'm going to write it out, but you don't have to know what this one is. This is a 4-carbon oxaloacetate. Oxaloacetate or oxaloacetic acid. This will pick up those 2 carbons from the acetyl group and make a 6-carbon citrate or citric acid. Okay, there's that citrate molecule that you guys have heard of before. We've talked about using citrate as a sole carbon source, and essentially it would be kind of a part of this process, not exactly, but this is the beginning of the Krebs cycle, and there are a number of other steps involved in the Krebs cycle, essentially, where there are reduction and oxidation events, or oxidation of the 6-carbon citric acid molecule to generate back that oxaloacetate. And as long as there's continuous input of this acetate here, or of this acetyl group here from this whole process or from some other process, then you can continue to have these turns of the Krebs cycle. So this is one turn of the Krebs cycle. Now there are some really, really key intermediates involved in the Krebs cycle that essentially either can be pulled out or, more importantly, can essentially start a turn of the Krebs cycle if acetyl is not available from this pyruvic acid or from other sources. So just note that there are lots of steps along the way that are very important, and I'll just write that down. For each of these acetyl groups, you're having one turn of the Krebs cycle, one turn of the Krebs cycle. All right, and so there are these 2 carbons that are coming in, 2 carbons with oxygens and hydrogens essentially are used, are donated to oxaloacetate to form citrate, and then they will be oxidized through these series of steps here. So some of these oxidations, the oxidations result in the loss of a carbon dioxide molecule. And there are 2 carbons in this acetyl group, so we'll have that as a loss of 2 CO2s for each of these turns here, and 2 CO2s for these turns here. We also have the donation of that acetyl group from that coenzyme. And remember, the function of these coenzymes is to essentially just continue to cycle in the cell, right? They just go back and forth from their holo form to their apoform. Oh, that's the opposite. Their apoform, their empty form, to their full form where they've picked up their substrate here, and back and forth and back and forth. So here is that recycling of that coenzyme A. There is ATP, there's ATP generated in this process, and I'm putting in the wrong color, that's okay. So we do have 1 molecule of ATP for each turn of the Krebs cycle generated through substrate level phosphorylation, kind of. Substrate level phosphorylation. Here and here, 1 ATP. And now let's draw our reduced electron carriers. So down here we have 3 NAD, H+, H+. Same here. We also have FAD generated in this process. 1 for each of these turns. 1 FADH2 generated here. 1 FAD, hopefully you're enjoying those chimes, FADH2 generated here. And that's it. There's lots of metabolic intermediates, but we're not going to write all of those in. Let's come back over here and draw our shorthand version. So again, understanding that there are 2 acetyl coenzyme A's that are generated from each one molecule of glucose, then we're going to say there are therefore 2 turns of the Krebs cycle for every input of glucose, right? We have a bunch of stuff that's generated from the end of this thing or as output from this process. So here, whereas we had 2 CO2s over here and 2 CO2s here, now we're going to just add all that stuff together and say, well, this is the complete oxidation of glucose, that 6-carbon compound. We saw 2 carbons lost here, and now the rest of them are lost here. So 4 CO2s. We have ATP. Whoops, sorry. 4 ATP. I mean, 2 ATP, I'm sorry. 1 for each turn of the Krebs cycle. So we have 2 ATP through substrate level phosphorylation. And then let's draw our reduced electron carriers. Again, we're just going to add it all up, what we wrote over there for our longhand version. So 3 plus 3 makes 6 reduced NAD molecules. And 1 plus 1 is 2 FADH2. Okay. So this is from your book. Again, that 3-carbon pyruvic acid. And remember that there are 2 of them for every input of glucose. So remember this happens twice. That 3-carbon compound is partially oxidized, where one of those carbons is lost as a CO2 to generate acetyl, right? This is picked up by a coenzyme A. So there's your acetyl coenzyme A. That acetyl group is donated to oxaloacetate or oxaloacetic acid to form the 6-carbon citrate molecule in this first step of the Krebs cycle. This initiates this turn of the Krebs cycle where there's a series of biochemical reactions that further oxidize this molecule to generate back oxaloacetate at the end of the cycle. And then this process can continue as long as there's continuous input of that 2-carbon acetyl group. Here's the full oxidation of glucose, or the complete oxidation of that molecule. We had lost 1 CO2 here, and then here's the other CO2s, right? So there's your 3 carbons that are lost from this pyruvate, and remember this is happening twice. There's that 1 ATP that can be generated through substrate level phosphorylation, plenty of metabolic intermediates that are produced here, and then our reduced electron carriers. Remember that this is for 2 turns of the Krebs cycle because of the 2 inputs of these pyruvic acids. So we're generating 2 ATPs, 6 NADH plus H pluses, and 2 FADH2. This is, if you're curious about each of these steps along the process, this is the actual biochemical makeup of this molecule, the 3-carbon pyruvic acid. Here's that 2-carbon acetyl group that's picked up by that coenzyme A, and that coenzyme A essentially just continues to shuttle as we drew in our diagram. This is that reduction event that's occurring as we have this oxidation or partial oxidation of this pyruvic acid. This acetyl group is donated to that oxaloacetate or oxaloacetic acid to generate this 6-carbon citric acid. And then we see this whole process here where we have a series of biochemical reactions. There are 1, 2, 3, 4 reduction events that are occurring, generating 3 reduced NADH plus H plus and 1 reduced FADH2. This is one of the key metabolic intermediates, alpha ketogluteric acid, and another one, succinyl coenzyme A. Note that under protein catabolism, this alpha ketogluteric acid is essentially generated from branch-chained amino acids, so they can be converted into this molecule here, and once they are, then you can at least have 1, 2, 3 reduction events occurring, and all of these reduced electron carriers can be donated to the electron transport chain to still generate plenty of energy. This is the concept behind, or at least one of the concepts behind, branch-chain amino acid protein catabolism. Again, you don't need to know all of this stuff. I'm just showing you for those of you who are a little bit more interested in the biochemistry behind this. This is what you do need to know. The yield for one input of glucose. So for every one glucose molecule, there are therefore 2 turns of the Krebs cycle. We have 2 ATP generated via substrate level phosphorylation. If you're a biochemistry person and you're curious about how this happens, you'll see it's a little bit different from what we just talked about for glycolysis, but for everyone else, we don't need to worry about the details there. For every 2 turns of the Krebs cycle, there are 6 NADH plus H pluses, 2 FADH2s, and then plenty of these metabolic intermediates that can be used for alternate processes in the cell, including that oxaloacetate and that alpha-ketoglutarate. So here's some questions for you. We did these discussions in class already. What's the final destination for electrons on those reduced electron carriers? You should be able to answer this one. You know that ultimately in aerobic and anaerobic respiration, they'll be donated to the electron transport chain, which is what we're talking about next. What's the starting compound that enters the Krebs cycle from glycolysis? Well, it's kind of a trick question. You have to answer the second part first. What transition step must occur? Of course, this is pyruvic acid or pyruvate, and it must be converted into an acetyl group and picked up by a coenzyme A in order for it to be donated to the Krebs cycle, or transition step if you want to make it simple. All right, so these things eventually will make their way down to the cell membrane in bacteria or the mitochondrial membrane for eukaryotic cells. And those electrons will be donated to the electron transport system or the electron transport chain. What is this? It's the concept here are that this is a series of electron shuttles or electron carriers that can be reduced and oxidized. And these membrane proteins essentially just pass these electrons down like a hot potato. So it's a chain of redox, reduction, oxidation carriers that receive electrons from reduced carriers like NADH plus H plus or FADH2 and pass those electrons from one molecule to the next. Now this diagram is showing you the potential energy input. So these electron carriers that are bringing in those hydrogen molecules, those 2 electrons and protons, have a high potential energy. Those electrons, once they're donated to those key players, those intermediates in the cell membrane or the mitochondrial membrane, as those electrons are passed on, essentially the amount of potential energy inside those electrons is reduced. And finally, you'll have the donation of those electrons with the addition of protons to oxygen to form water in aerobic respiration or an oxygen-containing compound in anaerobic respiration. So there is a reduction of energy as those electrons are floating, essentially, or passing from one compound to the other as you move along. And also note that the potential energy for NADH plus H plus is actually much higher than the potential energy for FADH2. And let's talk about these concepts here as we go through this. I'm not going to start with this picture, actually. Let's switch over to our whiteboard. Okay, let's try to draw this in the middle. I'd like to draw it here before we erase anything. So we'll go back up and put our black on. So all of these let me just highlight what we're talking about here. All of these reduced electron carriers that are generated here and here and here. And of course the FADH2 generated here. All of these guys are all going to make the cell membrane. And donate to the electron carriers that are embedded within that membrane. So here is our cell membrane. Actually, let me draw that a little bit bigger. And we have our embedded cytochromes and other types of electron carriers here. And downstream, I'll write this little kind of hat-shaped one. This guy is called the ATP synthase. So we're going to watch 2 different things, the destination of electrons and the destination of protons as they're shuttled through the electron transport system. So this is ETS, or electron transport chain, ETC. This is the periplasmic space out here. If you recall, this is the space between the cell membrane and the cell wall outside here. And this is, of course, the inside of the cell. All right, so we have our NAD. H plus H plus is always the starting compound here. These guys, and again, we're gonna look at the destination of electrons and the destination of protons. So I'm gonna draw our electrons in blue. So the first molecule in the electron transport chain will pick up these electrons, 2 of them. And these electrons will then be shuttled through these reduction oxidation events down and down and down. So they get shuttled all the way down the line from these electron carriers. Now, what's the point of this electron shuttling? And these guys eventually end with the donation to a terminal electron acceptor. Let's go back up. Again, what's the point of the donation of these electrons and the shuttling of these guys all the way down? Well, actually, the point is to push protons out of the cell. I usually do it in red, but I already have ATP in red, so let's do it in pink. So these protons have to go against their concentration gradient and get shuttled out into the periplasmic space. Now, how do they go against their concentration gradient? If you guys recall, what is that called when a molecule moves across the membrane against its concentration gradient? That would be active transport, and active transport requires proton transporters and energy. Well, for these protons to get shuttled out, there's no ATP input, but instead that push of these protons is powered by these reduction oxidation events. So what the function of the electron shuttle in the electron transport chain is to power what we call proton pumps. And so and I'm showing you kind of this thing just passes through, but that's not true. These guys cannot pass through. They pass through these proton pumps. So what I'm actually drawing here are proton pumps, okay? So this is facilitated by proton pumps. Proton pumps, that's what I'm showing you here. So these guys, some of them can act as proton pumps. Not all of them do, but many do act as proton pumps. So we see an example here. I'll draw another example of proton pump here and another one here, right? So these proton pumps can take free protons out here in the cytoplasm, or of course we have our other electron carrier, that FADH2. And the protons from this guy also get passed out. And so this is turned into FAD, and this can go back into cellular processes. Let's finish drawing our reduced and oxidized NADH. This is then oxidized into NAD, right? And this can then go back into cellular processes in the cell. FADH2, when it loses its protons, it also donates its electrons here, right? So those electrons can also get shuttled down. Now note that NADH plus H plus enters in way at the beginning of the electron transport chain versus FADH2 can also donate, but it donates downstream, right? So it cannot enter in the beginning of this. And so this is in part how we have the difference in the potential energy for each of these things. Again, the purpose of these electron shuttles, right, all of those passages of electrons, is to continue to power these proton pumps. And so out here, we're generating something called the proton motive force. Proton motive force, right? There's lots of protons that are generated here. Essentially, this is powering an electrical gradient, or an electrical gradient. So this generation, you can kind of think of this as like generating electricity to power these pumps to continuously push these protons out, much like you would see a water tower where water is pumped actively up into the top of the water tower and then can be released back down again with the facilitation of a transporter, right? So what is the purpose of this proton motive force? Well, the high energy force or gradient that has been generated is allowed to facilitatively be diffused back into the cell through this particular compound called ATP synthase. This is also called chemiosmosis, where you're allowing the diffusion of these guys back into the cell. So if you've heard of this term or you see it in your book, this is chemiosmosis. And this is where we have this ATP synthase allows essentially the force of this gradient to power the production of ATP, or the phosphorylation of ADP, to form that energy-rich compound, ATP. And this ATP is generated through oxidative phosphorylation, right? The use of reduction oxidation reactions to power the production of ATP, or oxidative phosphorylation. All right, one more thing to note here. I ended that showing you these transports of those electrons all the way down this electron transport chain, and mentioned that these electrons will terminate with a terminal electron acceptor. Now for aerobic respiration, this is oxygen, right? So a half of an oxygen compound plus two electrons plus 2 protons generates water. All right, so in aerobic respiration, oxygen is the terminal electron acceptor. For anaerobic respiration, this can be an oxygen-containing compound. This would be an oxygen-containing compound. An example that we've seen already would be nitrate converted to nitrite. Whoops, NO2. And this is done, of course, through nitrate reductase, but this is the terminal electron acceptor also, right? So this very last acceptance of these electrons would be nitrate to make nitrite here. Other types of compounds could be carbonates or sulfates. Where in all cases, all of these compounds can receive that donation of electrons at the end of the electron transport chain and allow that continuous production of ATP through oxidative phosphorylation by allowing that electrons to continuously shuttle through the electron transport chain. All right, so before we end this particular whiteboard session, let's do a quick count. Actually, I should mention this one first. Okay, so as mentioned, this NADH plus H plus has a high potential energy, so it can power the donation of those electrons to the starting compound here in electron transport chain, has the potential to power all of these downstream proton pumps, and all of these protons eventually that energy will be captured in this ATP synthase to generate ATP. FADH2 comes in downstream, so just thinking very simply about how many proton pumps these electrons here can power. Even in my diagram alone, you can see that NADH plus H plus has the potential to power 3 proton pumps just in this picture, versus the electrons from FADH2 only has the potential to power 2 of these proton pumps. And if the protons are what generates the force to produce ATP, then hopefully you can see how NADH plus H plus has the potential to generate more ATP through oxidative phosphorylation than FADH2 does. So because of this, we have a rule of conversion. And for every 1 molecule of NADH plus H plus, that equals roughly 3 molecules of ATP. For every 1 molecule of FADH2 that's generated, this has the potential to make roughly 2 molecules of ATP. Actually, yeah, I'm not even going to mention. All right, so this is our conversion rule, something that's really important for you guys to note for oxidative phosphorylation. So each of these NADs that are generated here, or NADH plus H pluses, has the potential to each of them make 3 molecules of FAD. I'm sorry, 3 molecules of ATP. For each of these FADH2 molecules reduced during the Krebs cycle, each of them has the potential to make 2 molecules of ATP. So if we recall, aerobic respiration has the potential to generate about 38 molecules of ATP, right? Aerobic respiration can generate about 38 molecules of ATP. Anaerobic is not as efficient, but still 32 to 36 molecules of ATP. But let's see if we can figure out where this number 38 comes from, okay? This is a combination of both ATP generated through oxidative phosphorylation and through substrate level phosphorylation. So let's do a quick tally of where we get our ATP from. I think I can fit it all on here. That's why I've saved a little bit of room at the bottom. Let's do our ATP count. ATP tally. Let's do glycolysis. We'll do our transition. And then we'll look at Krebs cycle and do our total. Okay. It would be easier for me to just do this on an Excel sheet, but that's okay. Okay, so let's do our NAD. Sorry guys, I'm not changing colors here. And then our times 3. How much FADH2 times 2. How much ATP through substrate level phosphorylation. And then we'll tally it all up in the end over here. In glycolysis, how much, and this is for every input of glucose is what we're talking about, how much reduced NAD is generated in glycolysis, going all the way back up here at the top. We see that for every input of glucose, we have the generation of 2 reduced electron carriers. Let's draw that in here. Okay, I'll change your colors for you just so you have them. For glycolysis, we have 2 NADH plus H plus that are generated here. How much for the transition step? Again, for each input of glucose, we have 2 pyruvates that are both converted into acetyl coenzyme. So that's 2 different reduction oxidation events. So we have 2 of these NADs reduced in the transition step. That should say 2. And then for Krebs cycle, we're turning this 2 times for every input of glucose. So we have 2 times of those 3 NADs. So we have 6 NADHs that are reduced to NADH in the Krebs cycle, right? And for each of them, we're going to multiply them by our conversion factor. So here, 2 times 3 makes 6. 2 times 3 makes 6. 6 times 3 makes 18, right? Let's look at FADH2. For FADH2, we have none. Where did my pen go? We have none in glycolysis. We have none generated in transition step. And we have 2 for every 2 turns of the Krebs cycle. There are 2 FADH2s generated in the Krebs cycle. So we have 2 here, and that's it. None and none. Again, let's look at our conversion. For every FADH2, there are 2 ATPs that can be generated. So here's times 2 equals 4. Now let's look for substrate level phosphorylation. For glycolysis, going back up, we have 2 net total generated through substrate level phosphorylation in glycolysis. So there's 2. For transition, there's nothing, no ATP generated through substrate level phosphorylation. And for the Krebs cycle, for every input of glucose, we have 2 turns of the Krebs cycle, and therefore there are 2 ATPs that are generated through substrate level phosphorylation. So for glycolysis, and there's 2 different ways you can tally this up, just go straight down or go straight over. Let's go straight over. For glycolysis, the combination of the potential ATP that can be generated from the reduced electron carriers, in addition to the ATP that are directly generated through substrate level phosphorylation. There's the possibility of generating 8 molecules of ATP through glycolysis. For the transition step, there's only reduced electron carriers here, so the potential energy is 6 molecules of ATP through oxidative phosphorylation downstream. And then for Krebs cycle, the majority of the energy is through potential energy that can be generated in oxidative phosphorylation, so 18 plus 4, 22 plus 2 that are already made through substrate level phosphorylation, that's 18, 19, 20, 24, 24 here. And we'll add all that stuff up. 24 and 6 makes 30, plus 8 makes 38. Okay, and there it is. There's your 38 potential molecules of ATP generated through all of these steps, the complete oxidation of glucose using the central metabolic pathways of aerobic respiration. Hopefully you got all that. Let's just go finish up these slides here for aerobic respiration, and then I'll do one more set of slides, or one more short video, hopefully, for fermentation and working through those alternate metabolic processes that are not using carbohydrates. So let's finish up these little videos or these slides here. And I want to stop here for a second because this is from your textbook, but I really don't like it at all because this really combines the concepts of protons and electrons together, and it really does not differentiate what's happening. But just so you see, a couple of things to note. First of all, that periplasmic space is the region between the cell membrane and the cell wall. This is an important thing to understand because any extracellular process or any chemical that can disrupt that cell wall will actually disrupt this. Not only will it cause potential lysis and with osmotic pressure, but importantly to note, this whole electrical gradient that's generated here, this potential energy, is destroyed if that cell wall is eliminated, right? So that's one of the other reasons why that cell wall is so important, is to actually maintain that chemiosmotic gradient essentially in that periplasmic space. So here though are those cytochrome electron carriers that are embedded in the cell membrane. Here's the donation of electrons to that first carrier and protons to the proton pumps from NADH plus H plus. Also downstream you'll have FADH2. Those protons are pumped out of the cell through to that periplasmic space with various proton pumps. And that proton motive force that's generated can be captured in that ATP synthase downstream, and that energy used to power the production of ATP from ADP. This picture down here is just showing you those terminal electron acceptors. And again, it really is not coupled with these protons here, but really focus on the electrons being shuttled through these cytochromes here, and those electrons being donated to these terminal electron acceptors, either oxygen for aerobic respiration or an oxygen containing compound like a nitrate or sulfate here for anaerobic respiration. Definitely, again, pause the video and go watch these videos from the lecture, the textbook resources that you have available for you on how this electron transport chain works. There are key compounds that are embedded in this, and actually we, for microbiology, oftentimes which particular electron carriers are in the electron transport chain helps identify, helps characterize the bacteria and give us IDs for them. So it's completely varied from one species to another. Even within the same species, they can have some variations here. But these are some examples of these electron carriers, these flavoproteins. There's some coenzymes there, and most notably cytochromes that have these metal atoms that at the center as cofactors that help facilitate these reduction oxidation reactions. I've mentioned already that proton motive force that is generated by those carriers that we just mentioned acting as proton pumps, many of them, and that generates that chemical energy gradient. We've just talked about how that energy can be harvested by that ATP synthase to generate ATP. But if you recall back from chapter 3, we also talked about how the bacterial flagella is powered by the proton motive force. So the redox reactions that push those protons out, those protons can be captured in alternate ways too, including the flagella and transportation of substrates. Again, if you have this available for you, I'm not going to show these videos within these lecture video overviews. I think it's a waste of our bandwidth time. but definitely pause again and go down to your lecture videos and watch how these proton pumps work. Okay. This is showing you the picture of this ATP synthase. I showed you guys this in class, and I definitely want you to go take a look at that video. I think it's in the next one, the next slide. This one, yep. So on your own time, as you have these lecture slides in front of you, open up that video or look at it on YouTube. That one's from YouTube. And watch this super cool electrical unit essentially capture that energy that's in that chemical gradient here and use that facilitated transport, allowing those protons to come back into the cell and actually using the mechanical energy in this really cool contraption to help power the production of ATP from ADP. And again, you can watch that on your own time. Again, energy yield, we just counted up how you got 38 molecules of ATP for every input of glucose in aerobic respiration. For anaerobic respiration, it's still a very high energy rich process, generating upwards of 38 molecules of ATP. And also hopefully an understanding of why these guys would switch preferentially if a cell can undergo aerobic respiration at any time possible, they will preferentially utilize glucose for that, because that is the high energy rich compound. So we just looked at this one. Why do electrons carried by FADH2 yield less energy, ATP, than those carried by NAD? And again, really focus on those proton pumps. And hopefully with that diagram we looked at, you can see how there are more proton pumps that NADH plus H plus has the potential to power than FADH2, simply because of where those electrons are donated into the electron transport chain. What's the final destination for electrons shuttled through the electron transport chain in aerobic respiration? Oxygen. And for anaerobic respiration, we know this can be an oxygen-containing compound. And then this last question, maybe it's last question. Where in aerobic respiration does oxidative phosphorylation occur? Only in the electron transport chain. How about substrate level phosphorylation? Just go back into your diagram and look through. That would be glycolysis and a Krebs cycle. Okay, I'm nervous that this video is going to get pretty long. So let me see how much time we have so far. We are going to pause this video here, and I'll pick back up again on the next video, just finishing up these slides. I'm nervous about how big these videos are going to get.